Hello, this is Jasmine Trigg and I'm a scientist at Sartorius. Today I'll be discussing a poster that I previously presented at the Federation of European Neuroscience Societies Conference, or FENS. And this poster was titled AI-Driven Image Analysis for Label-Free Quantification of Chemotherapeutic Cytotoxicity in Glial Cells. So a quick introduction. Glioblastoma multiforme, or GBM, is a malignant brain tumour that's associated with poor prognosis. To really progress effective chemotherapies, there's a need to develop more translational models and advanced quantitative methods, which allow us to get increased insight into cytotoxic effects in a non-perturbing manner. So here we're demonstrating how we can use the InKSight live cell analysis system and integrated software to assess cytotoxic effects of clinically relevant chemotherapeutics in glial cell types. For this, we use the InKSight AI Cell Health Analysis software module, and this is driven on pre-trained neural networks and is a robust solution for label-free cell segmentation and live-dead classification. So I'll show you across the next couple of slides some of the data, and this will really show how we can use the system alongside AI-driven analytics to assess cytotoxicity and its amenability to screening of therapeutic candidates. So the data I'm going to show you has been acquired using the InKSight live cell analysis system. And this is a fully automated high definition phase contrast and multicolor fluorescence system. And this resides inside a standard cell incubator for optimal cell viability. It's designed to scan plates and flasks repeatedly over time. This system exists alongside kind of a number of integrated solutions of both hardware as well as software, which I'll be showing today, as well as reagents and applications. And these are all kind of validated and really designed to work together. Our newest image analysis module is the InKSight AI Cell Health Analysis. And this software module needs very little user input, and the analysis is completely label-free. So we don't need any fluorescence dyes or reagents. And this really minimizes any disruption to our cells. So the workflow for this analysis is shown here. So after we've acquired phase contrast images using the InKSight, the images are then processed automatically using two AI models. So one will segment the individual cells, and then the other model will classify them as live or dead. And you can see some examples of how that is outputted in the image here. So we can see that the green mask indicates that the cells are live, and then the red mask indicates that they're classed as dead. We can visualize the data in real time, and the graph on the end here, for example, we can see the percentage of dead cells over time, which increases with higher concentrations of the cytotoxic drug. If we go back to the AI cell segmentation model, this was trained and validated on a wide range of cell types. And because of that, it can adapt very well to different morphologies, and some examples are shown here, predominantly just using some examples in some neural cell types. For example, we've got kind of A172s, or we've got T98G. It's also able to accurately segment kind of primary astrocytes that are slightly lower in contrast or more transparent, as well as non-adherent cells or semi-adherent ones, such as the BV2 microglia. So in this first figure, we're just looking at how we have kind of validated our AI-driven live-dead classification. So here, A172 or BV2 cells were treated with a concentration range of cytotoxic compounds, and we did this in the presence of InKSight Cytotox Green, and this is a marker of cell death. Cell death was then quantified using both fluorescence classification of the cytotox positive cells, as well as label-free AI cell health classification. We've got some example images in A here, so we can look at the phase and fluorescence rendered images for live and dead cells for each cell type. And the time courses show the percentage of dead cells in response to compound treatment. And we can see it's actual comparable time and concentration dependent responses between the fluorescence readouts and the AI cell health label-free readout. If we look at the concentration response curves, these confirm the analogous compound efficacies that we see using label-free and fluorescence analysis. Overall, this confirms that the label-free INKSIGHT AI cell health analysis is able to accurately identify cell death, and it can do this where cell death is induced by compounds of different mechanism of action and in different cell types. So to examine drug sensitivity, we compared 3 glioblastoma cell lines which had different PTEN expression statuses and looked at their response to chemotherapeutic compounds with varied mechanisms of actions. So initially we had A172, T980G and U87 MG cells, and these were treated with 13 clinically relevant chemotherapeutics, and we treated them with a high and low concentration of each in a 9-to-6-well plate. Images were acquired in the INKSIGHT over 3 days, and we quantified cell death using the INKSIGHT AI cell health analysis module. So if we look at the heat map on the left, this shows the percentage of dead cells at 72 hours for all compounds and cell types. And you can see that U87V shows reduced sensitivity for most compounds compared to the other 2 cell types. If we look at the representative images on the right here, these also show the phase images and the live dead classification masks for a high vinblastine treated cells. So we've got dead cells segmented in red and live cells segmented in green. So you can see that we've got kind of high adaptability of segmentation, and it's accurately classifying the cells across these different morphologies. And this data indicates there's differences in potency for vinblastine across the glioblastoma cell types. So the resulting data from the compound screen was used to select 4 compounds to investigate further based on observed differences in cell death across the different cell types. Subsequently, the 3 glioblastoma cell lines were sieved into 9-to-6-well plates and treated with 3-fold steroid dilution of cisplatin, Taxol, vinblastine, and doxorubicin. And then we monitored cell death over 3 days. The microplate graph on the left here shows the percentage of dead cells over 3 days for T980G cells in response to the 4 compounds and vehicle. The concentration response curves are shown for cisplatin and Taxol, and we can see cell type dependent differences in compound sensitivities, with U87 showing reduced potency compared to A172 and T980G cells for both of these compounds. The EC50 values and maximal percentage cell death for all of the compounds across the 3 cell types are shown in the table. So here we used a combined approach of live cell analysis and flow cytometry to examine cell death in a microglial cell line. The INKSIGHT AI cell health analysis module really enables label-free real-time monitoring of cell death and is non-exhaustive. So because of this, this really facilitates endpoint selection and allows for downstream experiments to be performed to probe deeper into mechanisms of cell death. So here we're using the IQ Human 4-plex apoptotic kit, which is a flow cytometry-based multi-parameter assay that enables quantification of 4 different apoptosis measurements per well, including caspase-3 activity, annexin-5 binding, cell viability, and mitochondrial membrane potential. And you can see the gating strategies that are used in BB2 cells here in A. So these cells are treated with 3 chemotherapeutics. This was using camptothecin, cisplatin, and carboplatin, all of which are known to cause DNA damage and activate apoptotic pathways. So we monitored these in the INKSIGHT with images being acquired every 2 hours. And then at 24 hours post-treatment, the cells are then harvested and transferred to the V-bottom plate. And then we labelled them with this IQ apoptosis kit. So as I've said, we've got the gating strategy here. In B, this is looking at the heat map of the 9-to-6-well plate. And here we're looking at the percentage of caspase positive cells expressed as a percentage of the single cells. And then we can see the transformed data at the bottom here. So this is looking at the kind of 24-hour CRC for the based on the InKSight analysis and the IQ Caspase readout as well. And you can see that we have comparable concentration response curves here for the two readouts. So to quickly summarize, live cell imaging enables acquisition of high definition phase contrast images and provides an ideal platform to study multifaceted biological paradigms in drug discovery. Using the InKSight AI Cell Health module, we can conduct unbiased and robust label-free segmentation and live dead classification of individual cells. Hopefully I've showed how the user-friendly software can provide high throughput, physiologically relevant insights into cytotoxicity in glial cell types and is amenable to the screening of therapeutic candidates. This label-free non-perturbing analysis is crucial for sensitive cell types and also enables downstream experiments such as high throughput cytometry to be performed. Overall, live cell imaging combined with advanced label-free AI-driven analysis is a powerful approach that provides objective, meaningful quantitative analysis of complex biological behaviour. Thank you for listening to this kind of short summary of the poster. If you'd like any more information, please visit our website where we have the poster available as well as a more detailed application note and more information on the relevant web pages.